Hello and welcome to the second video in the NCVE series of home science resources. We hope you really enjoyed the sensory test. I personally really enjoyed doing it. And if you haven't seen it, then you can find it on our website and also on the University of Reading YouTube channel, which we will give all details about that in the comments and in the PDF file attached to this video. So thank you again for tuning in today. Now today we are going to be looking at enzymes, all things enzymes, antioxidants. We're going to be looking at what an enzyme is, we're going to be looking at how an enzyme works, and then we're also going to give you the opportunity to learn about a specific enzyme that's really important in the food industry, and then go away and become scientists and run your very own enzyme test. And we're going to be doing that using potatoes or apples or bananas or lettuce, whatever you've got in the house out of those 4 ingredients, and some general household ingredients such as we've got some lemon juice, some bicarbonate of soda, some vinegar, some standard milk, and also the juice of an orange or any other fruit juice that you have in the cupboard. So nothing too difficult, and if you don't have any of these, then in the PDF file there's some alternatives that you could use depending on what you have. First things first, what is an enzyme? So if you're an older student, a GCSE student for example, and you've heard of enzymes, then pause the video and have a recap as to what you can remember the definition of an enzyme is. If you haven't heard of an enzyme before, then don't worry, we're going to be going through that in the next slide. An enzyme is a biological molecule that catalyzes, and by this we mean speeds up, the chemical reactions without actually being used up itself. They have lots of really important functions. For example, in the body, lots of enzymes help with the process of digesting all the food that you eat. There are enzymes in lots of things. There are enzymes even in your washing powder. They help to remove the stains. For example, proteases will help to remove the stains left by proteins. That is the difference between bio washing powder, meaning it contains enzymes, and non-bio washing powder, just like this, doesn't actually contain any enzymes. Enzymes and your digestion. Now, when you chew your food in your mouth, you are starting the enzymatic digestion process. So as you chew, you're breaking down your food into really small molecules, and then enzymes, some of which are in your mouth, some of which are in your stomach, will then start breaking down these smaller bits of food that you've chewed, using enzymes into really simple molecules that can then be absorbed into the bloodstream. So for example, if you were to eat, I've got an orange here. If you were to eat an orange, now an orange contains sugars, which are a form of carbohydrate. Now those carbohydrates are going to be producing simple sugars via the enzyme carbohydrases, which will act on those carbohydrates to produce simple sugars. Now, in a similar process, if you were to eat this loaf of bread here, now bread contains something called starch, and starch is acted on by an enzyme called amylase. So amylase will act on the starch and produce simple sugars. But bread also contains the carbohydrates, so you're going to have the carbohydrates working as well to produce simple sugars. Now, if you were to eat this bag of lentils, amongst other things, lentils are really high in protein. So here we have a different enzyme working, which are proteases. So proteases will act on the proteins, they will break them down into simple molecules which we call amino acids. Now here we have a pint of milk. So milk contains a sugar called lactose, and you may know of someone that is lactose intolerant. Now lactose is a sugar which is similar to the sugar that you would say use to bake a cake. It has a different structure, and it's found in dairy products such as milk and cheese. Now lactose has its own enzyme which is called lactase, and that breaks down the lactose sugar into 2 simple sugars, one of which is glucose and one of which is galactose. And finally, if you were to eat this cheese here, so the mozzarella, cheese contains both lactose and also fats. So to start with the fats will be broken down into fatty acids and glycerol, and that's via an enzyme called lipase, and that's because fats are called lipids. But also, cheese is a dairy product, so it contains lactose, so it's also broken down into glucose and galactose using the lactase enzyme. And we're going to be producing a further video on the digestive system, which may involve you getting a little bit messy, so if you've got a garden that would be great for this experiment, and we'll be going through the different enzymes that will be working through the digestive system and allowing you to see which parts of the body are involved. So how does an enzyme work? Well, now you know what an enzyme is, we need to know how it works, and we've given you some examples, for example, the enzymes in your digestive system. Now, an enzyme works on what we call a substrate. Now, a substrate is a substance that is specific for that enzyme, and when the enzyme and the substrate join together, we get a product. Now, you might be wondering what I'm talking about here, so I'm going to demonstrate with a little jigsaw puzzle. So if we take this piece of the jigsaw here, this is our enzyme. Now, the enzyme has a specific active site, which if you see this piece here, this is specific to the enzyme, which means that only a certain substrate or a certain substance will fit into that enzyme. So if we have our substrate here, and if we look here, they join really well together, and the substrate and the active site of the enzyme are compatible. So we have an enzyme and a substrate joining together, forming an enzyme-substrate complex. Now, we could have another substrate floating about, for example this one, and if you look when we try to join them, they cannot join. This is not compatible, and so this substrate is not specific for this enzyme, and we can't have an enzyme-substrate complex forming. So look again. We have it forming like this, and this is our enzyme-substrate complex, and from that we can get the formation of products, just like these ones here. So we could have this product coming off, and we could also have this product coming off. And if you look, the two products are exactly the same shape as the substrate, and they split and we get our products being produced. Now, we often call an enzyme and how it works as being a lock and key mechanism. If you think about it, your key is specific to your lock and your neighbour's key is specific to their lock. So you might be wondering, how does an enzyme, just like this one, know the shape of its active site? Well, an enzyme is a protein, and that means it can fold up in a certain way, and the way it folds allows it to be a certain specific structure, and that specific structure determines the active site and the shape of the active site, meaning that it is compatible for the shape of the substrate. So, what affects the action of an enzyme? Enzymes are affected by lots of things, but particularly temperature and pH. All enzymes will have an optimum pH and an optimum temperature, and that depends on where they work. For example, in the body, if you think about the enzymes that work in your stomach, their optimum temperature is likely to be 37 degrees, as that is the optimum temperature of the body. And the optimum pH of those working in the stomach is likely to be a lower pH because your stomach is acidic, compared to, for example, enzymes that work in the pancreas, which are likely to have an optimum pH of 8, as this is the pH of the pancreas. So it really depends on the environment that the enzymes are working in. Just a quick recap on the pH scale. So when we're talking about pH, we're talking about acidity and alkalinity. So acidic foods, these are your sour foods. They're at the lower end of the scale, they've got a smaller number. Your pH scale goes from 1 to 14. So for example, if we took this lemon juice here that we've been using, lemon juice is pH 2. And that's really acidic because the lowest end of the scale is pH 1. If we were to think of an apple is around pH 3.5 to 4, and a tomato, such as these chopped tomatoes here, are around pH 4 to pH 5. And we mentioned this in the last video, that low pH, which means we've got an abundance of hydrogen ions, which is what your tongue responds to, and that's why these foods, like the lemon juice and the chopped tomatoes, you could describe as being sour. So when we get to the other end of the scale, we're talking about alkaline foods, so anything from pH 8 to pH 14. Now, if we were to talk about this bicarbonate of soda here, that's around pH 9 to pH 10. And if we were to talk about this bar of soap, soap is around pH 12. And when we talk about alkalinity, instead of there being an abundance of hydrogen ions, we've actually got an abundance of hydroxide ions, so OH- ions, and that's what forms an alkaline food. And to put it into perspective, the glass of water that comes from your tap is pH 7, a neutral pH.